Hello everyone and welcome to this special episode of ESICM Inspiring Intensivist video series. I'm Burçin Halaşlı. I'm a consultant physician at Hacettepe University, Ankara, Turkey. I'm the member of Translational Biology section and as representative person in social media digital content committee on the behalf of Transbio section. It's a pleasure to be here today and an honor to guide you through this session with my colleague Belbria. The Inspiring Intensivist series was created with a simple but powerful idea: to share the stories, insights, and journeys of colleagues from around the world who are shaping the future of intensive care medicine. These conversations are not only about science or clinical practice, they are about inspiration, vision, and human drive to improve the care of our critically ill patients. Today's episode is brought to you by the Translational Bio section. Transbio connects the bench to the bedside and back, accelerating discovery through clinical relevance. We believe every ICU question deserves a molecular answer, and every lab discovery deserves clinical impact. Transbio section lies at the heart of ESICM vision for future-ready intensive care where science and practice meet. We hope this conversation will not only inform you, but also inspire you to think bigger, collaborate wider, and engage with Transbio community in shaping the future of intensive care medicine. I'm delighted to hand over the virtual stage to our co-moderator.
So good afternoon, and my name is Dr. Barbara McNicholas. I'm an intensivist at Galway University Hospital, and it's a real pleasure to introduce our speaker here today. So Manu Shankar-Hari is a professor of Translational and Critical Care Medicine at the University of Edinburgh and works as a consultant physician in intensive care medicine. Manu's research involves training in two disparate sciences. He has an MSc in epidemiology and a PhD in immunology. Manu's translational immunology research spans bench to bedside, focusing on dissecting clues from malfunctioning immune system to enable successful immunomodulation for critically ill patients. Manu leads a research lab focused on addressing one of the major global challenges in medicine and intensive care, that is sepsis. So with no further ado, I'm delighted to introduce the speaker, and I look forward to hearing what he has to say.
Thanks, Barbara, and thanks again, Burçin and the ESICM Transbio section for the opportunity to just discuss my journey as a researcher. And I've put together a few set of slides. Before that, let me acknowledge my funders to date. So most of my research funding is through UK-based research funders like the MRC program, the UKRI, HUO Foundation, and others. And I did hold an NIHR Clinician Scientist Award between 2017 and 2023. These are what I'm going to talk about as my views, not necessarily of the Department of Health. I do have some intellectual conflicts as highlighted by the person who introduced me, which is I'm a translational biology researcher. I run a precision medicine program and I chair the Lancet Commission on Sepsis. I don't have any financial conflicts to declare. What I thought I would do is pick a paper every year for the last 10 years and just introduce some of the different aspects of my work so that people can see that it doesn't have to be pigeonholed into one small bucket in terms of when you're training as an intensivist and training to be a researcher. So I think probably one of the most relevant papers in critical care, and one that I led, is the developing a new definition and the criteria for septic shock, and referred to as Sepsis 3 septic shock definition and criteria. It is a very simple idea, although there is quite a lot of methods behind it. It started off with a Delphi process, and then using some surviving sepsis campaign and other databases to come up with a concept of what we mean by septic shock. That's a definition. And then the concept was operationalized into criteria. If you look at the bottom half of your screen, you can see the different criteria that people had published in the literature about what septic shock is, and on the y-axis is the mortality. And what we ended up in the Sepsis 3 definition is hypotension plus need for vasopressor therapy and a lactate more than 2 as the criteria for septic shock. And why did we focus on that particular aspect? Because in the definition, we say that septic shock has a greater risk of death than sepsis alone. And I think that was essentially proven by the regression model that went with this analysis, where you see that when you do a risk-adjusted comparison, every other criteria had a significantly lower mortality compared to the criteria that we put forward. So in simple terms, it's you start with a description and then you ended up with a regression model to derive a criteria. So that's one of the papers based on the epidemiology skill set that I had. And this is a paper close to my heart, partly because when I trained initially as an epidemiologist and tried to get grants, and I was not very successful. So you start to think about what is it that you're missing in your science and how could you actually be a different scientist? And I think that's a reason for me going to do a PhD in immunology. So what this paper essentially talks about is B-cell biology in the context of sepsis, which is what my PhD is in. And the whole of 4 years I spent, when I was doing my PhD, we all knew that there is accelerated apoptosis. Richard Hotchkiss's work and others have shown that the patients with sepsis will have apoptosis. What my PhD and my research added is the first two words, which is activation associated. So before this paper, we thought that apoptosis happens, but we didn't know whether it was post-activation of a B cell or not. And my paper essentially addresses that. And the second thing that the whole of my PhD addressed was the fact that there is a differential apoptosis of memory B cells compared to in sepsis. Patients with sepsis often have an accelerated loss of memory B cells, and that has got importance in terms of vaccinating sepsis survivors. I'll come back to that right at the end of my talk. So what I showed here was using, again, flow cytometry, which is a basic science method, and then some intracellular staining to say that these cells that die in sepsis, that happens after activation. So an activated cell with greater energetic needs, but there is no energy, and therefore the cell goes on to die. So that was my kind of PhD work, and I'll come back to it because this eventually has led me 7-8 years down the line to design a clinical trial. Around the same time, I was interested in this particular aspect of sepsis, which is how do you know whether somebody who is coming to the emergency department has got sepsis if we were to do a white cell based assay. This was quite a complex study. Even though it involved only 6 or 7 centers, we had to standardize flow cytometry across different centers, and we were asking a question: what is the best biomarker to use? And those of you who follow sepsis literature, I think, will realize that there are probably thousands of biomarkers out there. And why is this paper important to highlight? And there are so many papers out there that I could highlight. So one, it is a paper that I led, but I think the thing that I really wanted to show here is this figure. We started off with roughly 47 biomarkers on a panel of neutrophil panel, a monocyte panel, a dendritic cell panel, and a lymphocyte panel. And then when you start to add methods, epidemiological methods to diagnostic biomarker kind of discovery and validation, you want a biomarker that is reliable, and that is what we tested in the second column. And out of the 47, about 40 are reliable biomarkers. And then we said, okay, if it is reliable, could we actually formally test that reliability? And when we formally test that, out of the 40, only 33 came out actually reliable in terms of inter-observer reliability. Okay, now that you've tested it, can you then do a cross-cohort comparison between 2 or 3 different cohorts and say which of the biomarkers still valid to carry forward? And out of the 33, only 17 were kind of valid enough to carry forward, and those were used for the discriminant analysis. And out of those which used for discriminant analysis, we found that only CD274 and 279, which are checkpoint molecule, and HLA-DR are valid biomarkers in the context of sepsis-related inflammation. And the reason why I'm putting this slide up is to highlight the fundamental point in biomarker discovery that we often overlook. Everybody publishes their favorite biomarker, and here I am showing you that actually out of the 47 favorite biomarkers that we had, only about 16 are reliable, and out of those only 3 are actually valid in terms of providing you with any useful information biologically. And I think that's an important lesson for me when I was developing as a scientist. That was kind of 2018, but it took around 4 years to do this work. And I got really interested in sepsis survivorship as I was developing as a researcher. And this was work that was funded through the National Institute of Health Research Clinician Scientist Award, focusing on sepsis survivors. Here it's again, going back to my epidemiology training, what we did here was to take the IGNAC database, which had roughly about 1 million records, and I think we found that about 95,000 or 100,000 records of sepsis survivors, and then we followed them up for a 5-year window using large-scale data linkage. And the research question here is, can you use those variables that are there at admission that we all know to see what is associated with their longer-term mortality? And I think this is useful, partly because everybody collects variables like age, sex, comorbidity, what sort of infection did they have, where was the site of sepsis, did they have septic shock, all of those things we all collect. And if these variables have prognostic information when a sepsis survivor leaves hospital, suddenly you can develop a score based on those variables. And I think that's what we did eventually. But this essentially shows that compared to age and sex-matched populations, sepsis survivors have a higher cumulative mortality over 5 years. I mean, if you look at it, that's roughly 4 times to 5 times higher mortality over a 5-year window. That's huge. It's worse than cancer if you are thinking of it, worse than heart disease. We're not doing anything for sepsis survivors. Again, I'll come back to that right at the end of my talk. And what we concluded is a very simple conclusion that actually, because we know there are certain generic features like age, sex, illness, severity, there are some sepsis-specific features like site of infection, presence of shock, you can create a score that then tells you which are the patients who are likely to do badly following their survivorship from hospital. And I think this is then COVID hit. We're into the 2020s now of my journey. And this paper although it was published in Nature Medicine with work that happened over a 4 or 5 month period. It's a bunch of other co-investigators alongside my co-senior investigator in the paper, which is Adrian Hayday, who's an immunologist. And I want to kind of give young researchers a couple of messages from this. So if you look at this paper, the flow cytometry paper that I showed before on, out of the 47 markers, only 3 are of any value. What we did in this is we have learned a lot about how to standardize flow cytometry quickly, and how to use multiplexing of flow cytometry and how to integrate flow cytometry data from that work. And I think that's what we did here. And this is, people weren't talking about systems immunology and integrating multiple domains of data when we actually published this paper in Nature Medicine. So here, if you look at the figure on the right, there are certain things that are essentially useful when you think about how to treat patients. The first thing I want to kind of focus on is the cytokine network at the bottom here, IL-6, IP10I, and IL-10. I'm going to show you a paper in the couple of slides where we suggested in this paper that actually IL-6 blockade might be valuable in the context of acute COVID-19, because it tells you not only that there is prognostic association, it tells you that the disease progression is higher if you have high IL-6. And as you know, IL-6 blockade I'll come on to in the next few slides. We also then showed in this work that TIM3 and PD-1, remember that I said CD274, which are checkpoint molecules, they are markers of exhaustion in the context of acute illness. And here we show again that actually these exhaustion molecules are significant and relevant in the context of COVID. So PD-1, L1, TIM3, these are T cells that are exhausted in the context of acute infection in COVID. And the final point I'd make here is the plasmablast on the right-hand side. These plasmablasts in patients who recover quickly with COVID were higher in our data. In other words, if you have a good plasmablast response that early, you recover quickly. And we also show that in our Nature microbiology paper, where how the trend over time changes if you have a good plasmablast response. And I then go on to kind of lead a convalescent plasma trial that is published in JAMA, where we don't show any improvement in outcome once you get critically ill. And perhaps you should be less ill if you're going to use convalescent plasma as an intervention in the context of COVID. And this is moving on to another paper again in the same issue of Nature Medicine that we produced. It was led by Michael Carter. This paper started off with a very simple observation. Kids were coming into hospital. They were COVID antigen negative. In other words, they were spike antigen negative. But these patients were very unwell. And they also had some sort of symptoms that lasted about 4 weeks ago, where they had an episode of fever-like symptoms, something like COVID, but nobody knew because they were kids, they got better. And then suddenly, 4 to 6 weeks down the line, these kids are coming into hospital and nobody knew why they were coming in. And ours was the first paper that essentially highlights an important point, which is you essentially have what is called as a new syndrome in the context of COVID. People were debating whether it's Kawasaki's disease and other sort of things, whereas we show for the first time that these patients had nucleocapsid and receptor binding and spike IgG positive. And these patients had very high CRP, which is T1 at admission, and T3 is when they come back for post-hospital discharge follow-up. And when you look at the cytokine profile, you can see that these kids at admission had, especially if they're antibody positive, they had high levels of inflammation. And they also had significant cardiovascular effects, like the NT-proBNP was very high, and that normalized over time. And when you look at the RBD antibody negative and antibody positive patients, you start to see a separation in terms of both symptoms as well as outcomes and coagulopathy. So this was the first paper, and I think we are quite proud of this because it's only 30 odd patients and it is Nature Medicine, and it kind of makes an important point that in the context of a pandemic, you can do very targeted science that answers an important question, which is these are kids with different disease, not Kawasaki's disease in the context of COVID. And this was picked up by NIH as one of those papers where they used that as a proof of concept funding for a lot of the COVID research that went in kids that went on in the US. So let me come back to the IL-6 story again. This I highlighted a couple of slides ago with our COVID IP, or the Nature Medicine adult paper. So around the same time, I got the opportunity to work with the WHO, partly as an epidemiology-trained immunology interested researcher in critical care. So they kind of asked me to coordinate a prospective meta-analysis for those young researchers beginning this space. And I think this is important skill to learn how to do a prospective meta-analysis. It essentially involves doing a systematic review of the trials that are ongoing. In other words, trials that are not completed and published, they are ongoing trials. You have to find out who they are and do a systematic review of the different trial registries like the clinicaltrials.gov and ISCRTN and other registries alongside the WHO registry. And this was essentially a project managed by the WHO, although I was kind of the lead author and the corresponding on this paper. It is project managed by the WHO with Janet Diaz as the main coordinator of all of this work. And it was collaborative. We had investigators across from around 30 odd trials in the context of IL-6 going on from the UK, US, Brazil, India, there are, and Russia. There are other countries that contributed to data to this work, and this essentially all of the protocol and everything was written even before the trials were completed, and that is most important thing because you want to avoid any form of bias here. And then you start to then do the analysis based on a predefined data set from all of these trials. And we show that about 11,000 patients who were randomized to either an IL-6 blockade or not. Could be usual care, could be placebo, but they didn't get IL-6 blockade. And what we show in this is that if you get IL-6 blockade, you had a better chance of survival. And I think the power of this sort of work is that it immediately gets translated to guidelines. And the day after, literally the simultaneous release was the WHO BMJ guidelines. And remember that if you are the investigator leading a prospective meta-analysis, you shouldn't be contributing to the guideline development because you are biased as a researcher. That's a principle we use. So I was in part of the guideline group, but I just led the meta-analysis. The guideline was published in the BMJ around the same time. So the key question here we asked is, what is the effect of IL-6 antagonist on an all-cause mortality in COVID? And we showed that it improves outcome, and it was adopted as the intervention across the world. So remember I said, I led the REMAP-CAP convalescent plasma trial, which was published in JAMA, and that trial was overall negative. I didn't put that paper up here just to highlight that overall trial, no difference between those who got convalescent plasma and those who got usual care. But in the UK LIMBO, the convalescent plasma domain of the REMAP-CAP trial, we had collected blood samples. And I think the hypothesis that we're testing here is, well, the overall trial is negative, and are there subpopulations that could have benefited or harmed from the intervention, because the overall trial is no difference between the giving convalescent plasma and usual care. So what we show here is a 3 subphenotype model, and the overall trial data is consistent with our subpopulation data. And more importantly, you see here that there is a treatment effect difference with the subphenotype 2 and 3 more likely to benefit from usual care rather than convalescent plasma. In other words, they were harmed potentially by giving convalescent plasma roughly. Although the confidence intervals crosses 1, partly driven by the fact that the sample size is smaller. We couldn't kind of pre-decide the sample size because we didn't know the treatment effect would be. So that's a sort of within a trial data is important for translation, because you could then explain why some patients benefit / identify a population that may benefit. Based on this, you would probably not give subphenotype 2 and 3 convalescent plasma. You worry whether you want to actually give subphenotype 1 convalescent plasma. Perhaps the treatment lies earlier on rather than critical care is what I would take home as a message. But those translational researchers training studies within a clinical trial is important. This is a paper led by one of the PhD students in the group, Rohit Saha, and the data was given to us by the Lung Safe Investigators, which is a study funded through the ESICM section led by John Laffey and colleagues. So what we did here is to ask a very different question. We had done the same, which I had done the same thing in sepsis about 8-9 years ago, where we all talk about sepsis trials being negative or no different, and you make a point that actually, if the treatment attributable mortality is much lower than you predicted, then your treatment may not work for that particular problem, because the excess risk by the disease is lower than you think, and therefore it's like an underpowered trial that you're doing in a crude way. So what we did here is to make a point by creating a propensity model for 4 different populations. So in ARDS, you compare that with patients without any hypoxemic respiratory failure. Then you compare ARDS patients with a single unilateral infiltrate acute hypoxemic respiratory failure, because ARDS is bilateral infiltrates according to the definition, and then you compare whether acute hypoxemic respiratory failure is different to non-acute hypoxemic respiratory failure. So those are the kind of different models in the paper, and that paper is in thorax. But I think the point I want to make here is slightly different, which is the final part of the figure, if you look at the subphenotype, the two subphenotypes that is published in the literature is hypo and hypoinflammatory ARDS. So if you got an ARDS patients, and if you got a non-acute hypoxemic respiratory failure as a control population, then the hyperinflammatory ARDS has got a very high attributable risk of nearly 60%. And if for a moment, take a population of patients with acute hypoxemic respiratory failure with a unilateral infiltrate, look at the attributable fraction now, much lower. And actually not different between the hypo and hyperinflammatory ARDS. And I think that's the sole point I want you to kind of take home. The whole idea that the ARDS-related syndromes, ARDS-related hypo and hyperinflammatory subphenotype have a very distinct outcome relationship only if you have bilateral infiltrates is so not true. You essentially have the similar sort of features if you have acute hypoxemic respiratory failure. We go on to show that in the paper. So in other words, you can design a clinical trial with acute hypoxemic respiratory failure rather than going down the bilateral infiltrate routes. That's what the kind of global definition comes up with, given the chest X-ray infiltrates are so not sensitive. So in the last few minutes, I just want to kind of talk about what's on the horizon. I started by saying that I've got a focus on sepsis survivorship, and one of the things that I said early on from a PhD is the accelerated loss of lymphocytes and loss of memory B and T cells, and when you recover from sepsis, the memory B and T cells don't recover. And memory cells represent your exposure to pathogens or vaccines as you grow up. So your episode of flu has got a flu memory, your DPT vaccine, your pneumococcal vaccine, or whatever other vaccines you've got a memory cell. And in sepsis, it takes that memory cell out. So one of the interventions that people have talked about, including me, is can we vaccinate these patients and see whether the outcome is different? But when you think about vaccinating sepsis survivors, remember we say that these patients have got high levels of T cell dysfunction. So, and do they actually respond to vaccines? Do we know? But we give vaccines. I mean, if you take a 60-year-old who gets a community-acquired pneumonia and goes home, when they are 65 or even earlier in some countries, they get a pneumococcal vaccine, they get annual flu vaccine, all sorts of things. Do we know actually whether that makes them better? Do they respond to the vaccine normally? When should we give them? Have we tested this question? So the simple answer is we haven't, and we just do it because we can. And I think one of the points that I wish to test is whether actually vaccinating benefit patients with sepsis, and could we do the sepsis survivorship vaccination in a much more kind of informed way. And I think that's the trial that we've just finished, and hopefully it'll be published in the next few months. So that's a vaccine trial that's under peer review currently. So that's in the horizon. And I think it also gives a story about the translational research problem. The idea started in 2014 to do a clinical trial of potentially vaccinating sepsis survivors. And I think the proof that needed to a funder for getting that vaccine was in 2017. And then it took me, the trial was going beautifully well in 2019 when the pandemic hit, and then we stopped. And I think for 3 years we couldn't do much, and then we restarted to complete the trial. Sometimes, translational research takes time, and I think that's a kind of an example of why I aged rapidly over the last 5 years. I think, so to summarize, the idea was to just give you a brief introduction to how I got here. And I've just picked up random work that I've done over the years, and I think I was asked to tell junior researchers what I would do, and I think that's my list, which is, I think, don't go after bright lights. I think you pick an area of interest and work at it. You need some mentors, and I've got lots of mentors, several of them, to just help me along the way. And I think I'll kind of stop there. And thanks again for the CChem Translation Bias section for the opportunity to kind of discuss this.
Manu, congratulations on a very inspiring journey and the opportunity to interview you about this. And I think your final slide has kind of trumped the question that I was going to ask, which is, how can you inspire intensivists to go down the translational pathway? Because I think over this talk, I think the word time, and this took time, kind of comes through. But without that time, we wouldn't be at a point where we can start deciphering the role of the immune system. And as you said, we need to do this if we want to improve the lives of our patients. So what are the strategies you think both the SICM should approach to really try and bridge the gap for intensivists? And would you advise your younger self to stay with the epidemiology, or are you glad you've stuck at the translational side?
So lots of questions there. So I think my lesson would be, I am a better researcher because I do have an epidemiological methodological training for sure. And I think the immunology PhD gives me a different dimension, and I think you kind of come up questions slightly differently. And I think that is useful both to have a different view and a different perspective, and it provides a different dimension to the team. At the end of the day, I think the days of small groups with single PI, king or queen, and then lots of worker bees, I think is probably gone. It's team science. You do contribute and lead your work, but it is team science. And I think the diversity of skill set helps you to build that bridge to do the team science. And I was fortunate to get a fellowship that let me do a clinical trial. So I think that's why I think I started off with a data science, epidemiology, health services research, and then did a PhD in immunology, and now I do clinical trials and biology within clinical trials. I think probably if you're trying to ask me a question, would I do something different? Probably not. I would do exactly the same training. It was hard. It was at times quite difficult. When you're a 40-year-old trying to write grants and nobody funds your grant, you just think, what the heck am I doing? And that's life. And I think sometimes I feel like, the translational bit helped me. So I wouldn't change it, I think.
That's good to know. And I am definitely running on mid-40s and still not getting funding. The other question I'll ask you, and it does come back to funding. So how do you convince funders to bridge this gap? Because a lot of funding agencies will only do basic science. Other funding agencies do clinical trials, and marrying in between is clearly what we need. So have you any thoughts on how we should approach that, or for people who are sitting down into the grant again, about how they can really try and at least get over the rejection maybe a little bit better?
Yeah, so it's an interesting question. So there is a fundamental issue here in critical care. I think one, we are a young specialty. I think we have to kind of acknowledge that there is lots more unknowns than knowns. We also are still learning smarter methods. I'm going to be slated by somebody, I'm sure, of saying that. So we still are, compared to a field like oncology or asthma, where they're probably 10 years ahead, and oncology is probably a bit longer than that, I think, in terms of how the specialty is developed. I think we are in that phase of trying to get there, and I think that's where the team science bit comes in. You essentially create a bunch of people with disparate skill set. You have to go after different grants for the same project of work. Like, for example, for a large randomized control trial that we are doing, we have a corresponding biology grant written to a different funder asking the question, there is a need to understand the mechanism by which this drug would work. We think this is the mechanism. We want to also understand, are there patients who benefit differently if the drug were to work? So there is quite a lot of focus on heterogeneity of treatment effect using EPI methods. There is an equally valid biological HTE question. In other words, if the drug has got multiple mechanisms, classic drug here, poster child is steroids. if you have multiple mechanism, there must be a heterogeneity of biological effect at a patient level. So that needs testing. That helps you refine who not to give the drug, which is adverse effect profiling based on the biology. So it's multiple grants. There is no escaping that unless you come with a very nice philanthropy funder who funds all of your life's work. It's hard to, you just need to find multiple. And I think the way to find multiple grants is not to lead everything by yourself. It's to be part of a bunch of people doing team science to lead different aspects of it. And that also is a huge opportunity for younger investigators, right? Because they could lead parts of the workflow whilst you're trying to develop your research profile.
Manu, I have a question that you drop here. I think the taking role models of is important for research, especially intensive care. If you think your journey as a puzzle, as a translational researcher, which piece is important for you that you decide on? Yes, I pursue my career on a clinical researcher. And what is the turning point, the most important turning point for you?
So I think the difficult question to pinpoint one event. I think there are a lot of events, I think. So one, as an example, as I said, I wasn't getting much grants just purely doing AP research. And I went and spoke to some of the funders to try and understand if they were to, not to the funding committee, but to members of the funding committee informally, to try and understand what it is in this grant that made the committee not fund it. And I think it's all well and good about talking about successful grants. Actually, you learn much more from the grants that you didn't get funded because you made some crucial error in the, aside from being unlucky, there's probably something missing. So learning from what didn't get funded is one important thing that I did. And then I think speaking to having mentors and speaking to them regularly is important. And I think they don't underestimate the value of informal conversations with mentors about where you could reframe some of the questions that you're posing. Often they don't need to be immunologists. They really need to be just clinicians or clinician scientists or a colleague who looks at it differently. And that conversations happen all the time. So I can't pinpoint one moment where that changed. I think that perhaps one big moment was me deciding, okay, maybe if I were to do a PhD in immunology, that might change the way I think about the topic. That probably was a pivotal moment. And I'm grateful for that opportunity that was given to me by my supervisor. She said, okay, give it a go, see what happens, what you can do with it, as it were. And that is important. And so there's a bit of opportunity, and I was lucky to get that opportunity, but equally, you need to be able to say, you're going to do a PhD with having a family and a daughter going to school, so you're all sitting at the same table reading the books in the weekend. I mean, that's not a great kind of advert poster child for family happiness, but you have to do sometimes that to try and get that bit of extra help to do your science.
For the last question, I want to raise emphasize the role of transbiosection. Well, what do you think the active and potential role of transbiosection in ISSCM for fostering the innovation and collaboration? What is your opinion?
So I think the translational biology section is, to me, it's a unique one because it kind of is, it straddles across the different other sections. If you think about infection, let's take an infection section and sepsis section or whatever. The underpinning science for all of that is the translational bit, and I think to me it feels like it's a cross-disciplinary activity, and it could involve multiple other sections as well as people who don't normally be an ESICM member. they don't necessarily need to be critical care researchers or critical care physicians. You could get non-critical care folk to be members of this biology section, because that's across disciplines. So there's an opportunity to kind of innovation within that space. So maybe something to kind of the society to think about, whether we can attract slightly different clientele to be members, to join particularly these sort of sections where diagnostic research and translation research is core to our future.
And thank you for your time and this just really incredible journey. I'd also like to thank Bersin for being my co-moderator, and the translational biology section and the team at the ESICM for facilitating this talk, and also, for facilitating this idea of team science. I think that's kind of one of the things I took from your talk. So I hope that it's inspiring for anybody who's listening as a translational researcher to listen to this journey and see how they can become part of it and improve the lives not of the patients we just see, but also for the lives that they lead in the future. So thanks very much.
Thank you. Bye.
Thank you. Bye-bye.